So I would now like to move on to Professor Dvorak. Professor Dvorak is the director of the Center for Vascular Biology at Beth Israel Hospital and at Harvard. And he was one of the discoverers of vascular endothelial growth factor. And he came up with a wonderful observation, which is that tumors behave like wounds that don't heal. I'm not going to give you more details about the individual laureates, as it's all in your nice little booklets. So, Professor Dvorak.
Thank you for that very kind introduction. What a great honor it is to be here today to speak to you and to have received the Gairdner Award along with Dr. Napoleon Ferrara. And what fun it's been to tour Canada. What a great country you have. What a big country you have. What nice people you have. Okay, this is my lecture outline. I want to talk about the discovery of what we call VPF, which is now known as VEGF, and some of the consequences of VPF-VEGF activity. I want to stress vascular permeability as an important part of the initiation of stroma formation that's required by tumors, wounds, and chronic inflammation. And I want to talk a little bit about pathological angiogenesis and something that has been neglected called arteriovenogenesis. And finally, I want to talk to the question, why doesn't anti-angiogenesis therapy work better in cancer? Napoleon has shown that it works so very well in macular degeneration. Okay, the story begins long ago, back in the 1970s, when my wife and I, here pictured, we were immunologists, and we were studying an interesting reaction, an immune reaction called cutaneous basophil hypersensitivity. And it was a form of cellular immunity in which large numbers of basophilic leukocytes accumulated. And you see the leukocytes that are pictured there. And this is what an electron micrograph of a guinea pig basophil looks like. So we thought that we performed this reaction with a variety of antigens, protein antigens, viral antigens, and so on. And we thought it would be interesting to see what tumor antigens did. And this was my eureka moment. It's fun to have eureka moments. There aren't many in life. And in the left-hand panel, you see what we saw. We saw a clump of tumor cells in a freshly transplanted cancer. But there's all this clear space around, and the arrows point to what turned out to be fibrin. And fibrin was deposited in these solid tumors, and it took the form of a gel. This is what fibrin looks like in a test tube. And as we looked at tumors by immunohistochemistry, human tumors, animal tumors, they all seem to have deposits of fibrin present in them, as shown in the slide. So how does fibrin get into tumors, into solid tumors? Well, for that to happen, two things ultimately have to happen. Tumors don't make fibrinogen, the precursor of fibrin, and so this fibrinogen is a plasma protein circulating in the blood that normally does not get out of blood vessels. And so for fibrin to form outside of blood vessels, the vessels have to be leaky. That's postulate number one. Not only leaky to fibrinogen, but also to a variety of clotting factors associated with the clotting mechanism. And of course, the second thing that has to happen is clotting has to take place in the extravascular space. So does this apply to tumors? Well, yes, it does. If in the left-hand panel, if one injects a fluorescinated dextran, a large molecule, one sees that here it is in normal-appearing blood vessels, but here the arrows indicate hotspots where there's been considerable vascular leakage. So in this tumor, and in really all tumors that have been examined, human and mouse and guinea pig and so on, increased permeability is a feature. So that's the left-hand panel. The right-hand panel shows that tumor cells also induce clotting. They induce the extrinsic clotting pathway because they express tissue factor on their surface. Further, we showed that tumors shed microparticles, or exosomes as they are now called, small membrane-bound vesicles that also express tissue factor. And in this manner, the clotting ability of the tumor cell could be spread as the exosomes spread around it. So both tumor vessels are leaky and clotting takes place. So clotting is not unique to tumors. If you cut your finger or cut yourself or bleed in any site, you're going to bleed to death unless you activate the clotting system in the extravascular space. So we figured that it would be more interesting to study the permeability activity. And what we postulated was the tumors secrete a unique vascular permeability factor, which we call VPF, that degranulates mast cells or basophils releasing histamine. We've been studying basophils and histamine and mast cells. Those granules that are shown in these electron micrographs contain a lot of histamine. And when the basophil here degranulates in this fashion, these granules are shed into the outside space, and histamine is released in large amounts. So we tested that hypothesis, and as often in my experiments, the hypothesis was wrong. In fact, we found in tumor culture supernates that there was an activity, a vascular permeability factor, that induces permeability to plasma proteins, and it acted selectively on venules. There was none, and you can see with the primitive methods we had in those days for analyzing the protein, we showed that it was a protein. But it didn't degranulate or release histamine from mast cells or basophils, and antihistamines did not affect its activity. So our hypothesis was wrong, like many of my hypotheses. And so VEGF is acting directly on endothelial cells. So Donald Sanger, who was a postdoctoral fellow in the lab at that time, purified the protein through traditional column chromatography, purified it to homogeneity, demonstrated that it was an N-glycosylated dimer, and it had tremendous vascular permeability activity, some 50,000 times that of histamine. It acted directly on vascular endothelium, as I've shown. The assay we used, which you may get a chuckle out of, was this, called the Miles assay. And what is done in this assay is you inject into a guinea pig skin or into a mouse skin a small amount of the activity, the material you think has permeabilizing activity, and you inject a blue dye intravenously, a blue dye which binds to plasma albumin, and so wherever the plasma albumin goes, a blue spot appears, and so you can see a blue spot was a positive column fraction. We also found that most malignant tumors, whether human or mouse, express this activity in large amounts using in situ hybridization, which is shown here. So what about fibrin that forms in these tumors? Is it of interest or not? And it turns out that it's an active player in stroma formation in a number of respects. First of all, it's a marvelous substrate for cell migration, and that's illustrated in the upper left-hand panel, where one sees fibroblasts migrating in a fibrin gel. And this is interesting because cells of a variety of different types all like to migrate in this fibrin gel. It's a promiscuous matrix because it binds to integrins expressed by many different cells and allows cells to migrate through a fibrin gel, much as children in a jungle gym migrate through a jungle gym, attaching and detaching. But I think of more interest is the fact that it by itself is able to induce angiogenesis and mature connective tissue formation. Now, tumors have to induce a stroma if they're going to survive. I sometimes ask medical students, what is the stroma for liquid tumors, such as leukemias? And of course, the answer is the plasma itself. That is the ideal stroma for a tumor that supplies its nutritional needs and also removes waste products. But solid tumors have a tougher problem. They have to induce a stroma. And that stroma consists of new blood vessels and fibrous connective tissue, which replaces the fibrin. And interestingly enough, if one simply makes a fibrin clot in a test tube and implants it in a mouse or a guinea pig, one sees what happens in the right-hand panel, where the fibrin on top is over time, over the matter of a few days, is replaced by fibrous connective tissue, vascularized connective tissue. So fibrin, in some way that is still not fully understood, has this ability to amplify the angiogenic response. So, I'll now call it VEGF, initiates tumor stroma generation in a characteristic sequence of events. Increased vessel permeability, fibrinogen and other plasma proteins leak, extravascular clotting, fibrin deposition takes place, and then the twin processes of angiogenesis and provisional provisional stroma is replaced by mature vascularized connective tissue stroma. Well, my hypothesis was that tumors were making a unique substance. Of course, that turned out to be wrong as well, because in fact, what tumors do is simply activate the wound healing process. Within just a few hours of wounding, as shown in a skin wound in the upper left, there's a great increase by the epidermal cells of VEGF. It's made in large amounts within a very few hours. The blood vessels become leaky, and one has increase in vascular permeability, fibrin deposition, and vascular permeability increase. The difference is that in the case of a tumor, this vascular permeability persists because VEGF is made continually, whereas in the case of a wound, as the wound heals, the hypoxia initiated by the wounding itself disappears, and VEGF production decreases. So tumors, in fact, appear as wounds that do not heal. But it's not only wound healing that this, and I would add that this is a skin wound is one example, but myocardial infarcts or strokes are other examples of this very same process taking place. But it's not only wounds that are involved in this process. It's a variety of chronic inflammatory diseases, such as rheumatoid arthritis, illustrated in the right-hand panel, where one has fibrin deposition on the pannus, overexpression of VEGF, and overexpression of the VEGF2 receptor, which is the most prominent receptor for VEGF. So I'd like to have a brief aside here on vascular permeability, because it seems to be a subject that is not well understood. First of all, vascular permeability is a very important thing. For example, consider the kidney, where large amounts of VEGF are made by podocytes, and large amounts of permeability take place through the kidney every day. But it's a little different from tumors and wounds in that this basal vascular hyperpermeability, first of all, occurs in capillaries, but it's also a filtrate of small molecules, very little in the way of plasma proteins. In contrast, in pathology, one has a plasma protein-rich exudate, and it occurs in two fashions. First of all, it occurs in something called acute vascular hyperpermeability, and this is what happens when you get a mosquito bite and you have a short-term release of histamine. And this occurs not in capillaries, but in venules. So one has arteries and arterioles feeding a vascular bed, a capillary bed, and here's where basal vascular permeability takes place. And then in venules is where acute vascular hyperpermeability takes place. But of probably greater interest is something called chronic vascular hyperpermeability, and this is the long-lived hyperpermeability found in tumors and wounds and chronic inflammation. And no normal blood vessel is responsible for this type of leakage. Rather, a new kind of blood vessel is formed primarily from venules called the mother vessel, and that's illustrated in the right-hand panel. So just to recap, basal vascular permeability takes place in capillaries. An electron micrograph of a capillary is shown in the upper right-hand panel. And you can see that within the cytoplasm of these capillaries are large numbers of caveoli, an organelle discovered by George Palade many years ago. And you can see that it's a very thin squamous epithelium. And the filtrate, as I mentioned, is mostly small molecules. And the small molecules get through primarily through pathway number 1, illustrated on the slide, that is between endothelial cells. But you do need small amounts of protein in the stroma, and this occurs through these caveoli that Palade discovered. And he originally postulated that these caveoli bud off from the luminal surface, carrying a cargo of proteins, of course fluid as well, and migrate across the cell where they discharge their contents at the abluminal surface. And of course, they can sometimes link together to form a chain of vesicles. But acute vascular hyperpermeability is a little bit different. And as I mentioned, it takes place in venules. And here's what a normal venule looks like by electron microscopy in the upper left-hand corner. And you can see in the yellow circles large clusters of vesicles and vacuoles. And this is a characteristic feature of venular endothelium. Venular endothelial cells are cuboidal. They're taller much taller than capillary endothelium, as you can see here. Here's capillary endothelium. Here's the much taller venular endothelium. And you can see that the vesicles and vacuoles seen by electron microscopy, and here illustrated in a diagram, consist of multiple, indeed hundreds of attached vesicles and vacuoles that are attached to each other and that extend from the lumen to the vascular ablumen. They interconnect at stomata, which are covered normally covered by thin diaphragms. And on the right-hand panel, you can see a reconstruction, a computer-based reconstruction of these vesicles and vacuoles. In acute vascular hyperpermeability, these vesicles and vacuoles, this VVO structure, opens to allow the passage of fluid and proteins, as illustrated in panel number 3. Now, there still is controversy in this area, and many people also advocate that pathway number 4, that is in between endothelial cells, takes place. Certainly, if one looks at tracers at the electron microscopic level, as done by my wife, Ann Dvorak, one can see, especially with HRP, that within 10 seconds of injection of HRP, one sees a reaction product that extends in vesicles and vacuoles and comes out at the bottom here. At this time, there's no reaction product in the endothelial cell junctions. So certainly the transcellular pathway is effective. But now in pathological angiogenesis, one has chronic vascular hyperpermeability, and this is the chronic hyperpermeability of tumors, wounds, and chronic inflammation. And I'd like to talk about that for a moment. And in doing so, I'd like to mention Judah Folkman, who in 1971, actually at our hospital in Boston, made certain postulates that are listed on the left-hand panel. He said that tumors must induce new blood vessels to grow beyond minimal size, that they made a tumor angiogenic factor, a TAF, that was responsible for this, and that anti-angiogenesis should be effective therapy. There should be a multiplicity effect, that is, killing one blood vessel should kill many, many tumor cells. Tumor vessels are abnormal, so they should be targetable. It should be applicable to many cancers, because if TAF is made by a variety of cancers, attacking it should be effective therapy. And vascular endothelium is highly druggable. It's accessible to circulating drugs, antibodies. Drugs don't have to pass through the endothelium to get to the target. I should also mention one postulate that was not made by Dr. Folkman, but by Bob Kerbel, who's in the audience. And he postulated that this type of therapy should be very effective because it should not achieve resistance to therapy, as, for example, chemotherapy does. So where are we in 2014? Well, Folkman's postulates have been largely validated. Tumors generally do need to make new blood vessels to grow beyond minimal size. They do so at least initially by secreting a VEGF, and that turns out to be Folkman's TAF. Tumor vessels are indeed abnormal, and so they should be potentially targetable. And anti-VEGF therapy has indeed found a place in treating human cancer, largely due to Napoleon's work. But the promise of this type of therapy has not been realized in human cancer. If you look at anti-angiogenesis therapy, first of all, it's mostly anti-VEGF therapy or targeting the VEGF receptor through receptor tyrosine kinases. Various drugs are listed there. And it prevents very effectively the growth of most fast-growing transplantable mouse tumors, and Napoleon showed this many years ago. Unfortunately, it works less well on mouse tumors with an established vasculature and has limited effectiveness in human cancer. And the famous slide of Herb Hurwitz is shown at the bottom there, where a combination of chemotherapy plus bevacizumab, the antibody against VEGF that Napoleon developed, causes an increase in survival in colon cancer by about 4 to 5 months. And this type of therapy is used now in a variety of cancers. So it's a good time to ask some basic questions. What are tumor blood vessels? How do they form? And why doesn't this anti-VEGF therapy work better in cancer? Well, I can deal with the first two questions fairly quickly because this is work that we've long published. First of all, tumor blood vessels are heterogeneous, of at least 6 different types. Often one thinks of angiogenesis as if it were a single type of blood vessel. This is not the case. It consists of at least 6 discretely different types of vessels that differ in structure and function. And most of these vessels are quite abnormal, differing significantly from those found in any normal tissue. And in addition to angiogenesis, which refers to the development of new small vessels from pre-existing small vessels, there's also something called arteriovenogenesis. And I want to emphasize this because I think it's very important. And here it is. If one looks in the left-hand panel at a mouse tumor, and it happens to be growing in the ear, but what I'm saying applies equally to human cancer, within this yellow border is where the tumor is, and here's where all the angiogenesis is taking place. However, if one looks outside the tumor mass, one sees very abnormal new blood vessels forming, large vessels, new arteries and new veins. And this is arteriovenogenesis, which has really received very little attention. And these vessels, as you can see here, are extremely abnormal. Okay, the first question was, what are tumor vessels? The second question now is, how do tumor blood vessels form? And I think it's clear that VEGF is the primary factor that causes the induction of new blood vessels in tumors, wounds, and chronic inflammation. Studying tumor vessels, however, in tumors is very difficult because they comprise only a very small component of the tumor mass. So some years ago, we reasoned that if tumors are making VEGF and inducing new blood vessels in that fashion, then expressing VEGF in another way, without tumor cells, should be an effective way of finding large numbers of these vessels so that one can study them. And so what we did is we expressed VEGF in an adenoviral system. And when VEGF is then injected either into the skin or into the ear, for example, in the top panel, subcutaneously in the bottom panel, or into a variety of other tissues, one really gets the same sequence of new blood vessels forming. And you can see how it proceeds from day 1, day 3, day 5, day 10, and even out here to day 128, one has very large numbers of new blood vessels that are formed. Similarly, in the lower panel, the same sequence of events takes place when the adenovirus is expressed in the subcutaneous space. And I think it shows nicely in the yellow circle here, you can see where all the angiogenesis is, and the blue arrows point to the arteriovenogenesis. So similar to tumors, the anti-VEGF system models the tumors. So both angiogenesis and arteriovenogenesis proceed linearly and sequentially over time in the fashion shown here. VEGF-A is expressed whether by a tumor or by an adenovirus, and one gets angiogenesis in this top panel where venules and capillaries initially form mother vessels and then a series of daughter vessels that form. And in the lower panels, one has arteriogenesis and venogenesis resulting from the remodeling of pre-existing arteries and veins. The first angiogenic vessel to form is the mother vessel in the red circle. So what is a mother vessel? Well, it's a very large vessel that is formed from pre-existing venules, primarily. In this light microscopic view, one goes from a vessel of this size to a vessel of this size, and one sees detachment of pericytes. Remember that small blood vessels have 3 layers: lining endothelium, then basal membrane, and then pericytes. All 3 layers are important. By electron microscopy, again at the same scale, one goes from a normal venule of this size to this very enlarged mother vessel engorged with red blood cells and a very thin endothelium with detached pericytes. And these cells are dividing cells. So how does this take place? Well, it forms by a 3-step mechanism. First of all, and most important, is proteolytic degradation of the basal membrane. Vascular basal membranes are very rigid structures and do not allow the expansion of a vessel beyond about 30%. So if you're going to get this 4-fold or 5-fold increase in cross-sectional area, one has to have a degradation of this basal membrane. And we found some years ago that the proteases responsible for this are cathepsins of various types, and also that there are what are called CPI, cysteine protease inhibitors that normally inhibit cathepsins. And whereas the cathepsins go up, CPI go down. And so that's the very first step that takes place. Now, pericytes normally attach to basal membrane and penetrate the basal membrane to contact endothelial cells. But when the basal membrane is digested, these endothelial cells detach, the pericytes rather detach, and therefore one ends up with a blood vessel that is lined only by endothelium. And one then gets a 4 to 5-fold vessel enlargement as the basal membrane restraint are removed. And hydrostatic pressure is what drives this. One goes from this normal venule to the hyperpermeable venule of AVH to this very thin endothelial cell structure as pressure within the blood vessel drives this increase. And the arrows that are pointing to here, as these attachments disintegrate, one gets this very thin vessel. As VVO membranes, the membranes lining these VVO structures migrate to the cell surface, providing the extensive new plasma membrane that is required for mother vessels. And mother vessels are the only type of blood vessel that is permeable to plasma proteins or other macromolecules, shown here with 3 different tracers. So one has 6 different types of new blood vessels that form in tumors, but only one of these is responsible for the vascular permeability. So the third question I wanted to address was, why doesn't anti-VEGF therapy work better in human cancer? And there are many possible reasons for this. Possibly one needs to treat earlier, better dosing, and I think a lot can be done in this area. Remember that we are treating in patients are elderly, they have other complicating factors, whereas mice are young and healthy, the ones we study for tumors. So I think a lot of improvements can take place. We also have to remember that by its very success, or partial success, ischemia induced by anti-VEGF drugs allows for the increase of VEGF and other growth factors, which are stimulated by an ischemic environment. And finally, I suppose there's the possibility, which we'll discard immediately, that targeting tumor blood vessels are of limited value. Nobody believes that, right? But there are 3 other possibilities, however, that do need to be considered. One is that tumors co-opt pre-existing blood vessels. In other words, they make use of vessels that already exist. Another possibility is that tumors survive hypoxia, and they live in a hypoxic environment, and they need only few new blood vessels to survive. And the third possibility, and one we've studied a lot, is that not all tumor blood vessels are susceptible to anti-VEGF therapy. So let's consider these 3 possibilities. Well, possibility number 1 is that, for example, lung metastases, one has cooption. In other words, the tumor is making use of pre-existing blood vessels. And certainly in all the tumors that Bob Kerbel and I have looked at, this seems to be the case. In this panel, upper left-hand panel surrounded by red, one can see tumor cells living in the alveoli of the lung. They've metastasized there. And they're perfectly happy because they have all the oxygen they need and nutrients supplied by the blood vessels that are present abundantly. But no new blood vessels have formed. And this also occurs in human cancers. Here, for example, is a human adenocarcinoma, and in the reds, surrounded in red, are individual clumps of tumor cells in the lung. I don't want to leave you with the impression, however, that this is the most common event. Very often, human tumors in the lung do induce angiogenesis. So that's possibility number 1. Possibility number 2 is illustrated below, where if one looks at the histology of breast cancers or pancreatic cancers, one finds a very abundant collagenous stroma, and there are actually very few blood vessels present. But there's a third possibility, and that is anti-VEGF therapy affects only subpopulations of tumor blood vessels. As I mentioned, tumor blood vessels are heterogeneous, 6 types. They form linearly over time. Early vessels evolve into late vessels. And in mice, anti-VEGF therapy prevents very effectively the growth of early tumors, but it's less effective in tumors with established blood vessels. And of course, in humans, this is the case because human cancers are not detected for months or years after they originate. And so blood vessels have been forming for a long time, and presumably a majority are of the late type. So the hypothesis we put forward is that early vessels are VEGF dependent, whereas late vessels, although they were induced originally by VEGF, have lost their dependency on tumor VEGF, not susceptible to anti-VEGF therapy. Notice I said dependency on tumor VEGF. And in experiments that I think demonstrate this point, we've looked at aflibercept, anti-VEGF therapy, antibodies, receptor tyrosine kinase, rapamycin, and all of these therapies target early blood vessels, not late blood vessels. For example, the way this experiment was done is that we used an adenovirus expressing VEGF-A and administered it on day 0. And then treatment was given, as shown in the left-hand panel. Group 1 received therapy on days -1 and +2, harvesting the tumor at day 5. Group 2, again, anti-VEGF at day 0, treatment on days 5, 8, and 11, harvest on day 12, and so on down the line. In Group 5, for example, treatment was delayed till day 51. And one can see that in the top panel, one forms a sequence of events that is shown here. Group 1, day 5, one gets vessels that look like this. Day 12, day 21 and here out at day 61. Sequence of events that are very abnormal vessels persist out here at day 61. Treatment results in very effective therapy at these early times, but at later times there is essentially no effect. And this is illustrated quantitatively in the slide in the lower left-hand panel, where blue indicates leakage and red is intravascular volume. And one can see that in controls, one gets a lot of leakage and a lot of new vessels, whereas the treated animals are very effectively inhibited. But the treatment is progressively less effective as one goes out in time, and finally at day 61, one has little or no effect, even though one has a doubling of the intravascular tumor volume. So to summarize, all of these drugs are very effective at preventing anti-VEGF-induced angiogenesis and arteriovenogenesis. They regress early vessels. They have little or no effect on late blood vessels. And mother vessels, which are the great majority of vessels in susceptible mouse tumors. This predicts the limited effectiveness of anti-VEGF therapy on human cancer. And the susceptible vessels are located within this blue dashed box. So where do we go from here? How can we improve anti-vascular treatment? We can't do much about cooption or tumors that can survive on limited vasculature. You can target additional growth factors and receptors besides VEGF, and that's a viable approach. Or, as I'm hoping to do, find new vascular targets. And if you were a plumber, what vessels would you try to attack? Would you attack the feeding arteries, the angiogenic vessels, or the draining veins? Current therapy attacks these angiogenic blood vessels. But if you were a plumber, you would not want to go and have problems with plumbing in your house and you wanted to turn it off, you would probably go to the point where the water system enters the house. And you'd probably do that rather than trying to turn off the faucets in every individual, in the kitchen and every individual room of the house. So that's what we're trying to do, and we'll see if we're successful. I don't know yet. Our strategy is to use this anti-VEGF system. One can isolate these vessels in very pure form and screen for targets and see if we can find some targets that will be effective. So take-home message, much too long. All tumors have stroma. Tumors generate new stroma and blood vessels by secreting VEGF. Vascular permeability has a key role in initiating stroma formation. Fibrin is the initial provisional stroma, has a role in generating mature stroma. Tumor stroma formation mimics wound healing and chronic inflammation, invented nothing new. Tumor blood vessels are heterogeneous. They derive from two distinct parallel processes, angiogenesis and arteriovenogenesis. Mother vessels are the first new angiogenic blood vessel to form. They're the subset of vessels that's hyperpermeable. Anti-VEGF is useful in preventing angiogenesis, arteriogenesis, and regresses early but not late blood vessels, and so new vascular targets are needed. Now, I've mentioned others who did the work, but this is my best collaborator, my wife of 52 plus years. And these are our three best experiments. We did not get the Gairdner Award for this. And these are some of my heroes. Thank you very much.